The human body is a chaotic environment. Every individual possesses a unique genetic makeup, distinct environmental exposures, and varying behavioral habits. The primary challenge of medicine is finding a universal truth, a treatment that works predictably across that immense biological diversity. We start by looking at what is already happening through clinical research. In observational studies, researchers act as bystanders. They record behaviors, symptoms, and outcomes over time without interfering. Methodical. This approach is excellent for identifying associations, like noticing that a specific diet correlates with lower rates of heart disease. But observation alone has a strict limitation. It cannot definitively prove causality. There are simply too many overlapping factors in a patient's life to know if the diet actually caused the improved health, or if it was something else entirely. To prove exactly what causes an outcome, researchers must use experimental studies. Here, the scientist deliberately influences events, applying a specific intervention, like a new drug or surgical technique, and recording the exact effects. The absolute gold standard of this approach is the randomized controlled trial, or RCT. Observational studies point us toward where we should look. Experimental trials are the engineered tools we use to prove what actually works. Before an experimental trial can prove anything, it has to conquer its primary adversary, variability. This diagram illustrates the problem. In a controlled classroom, a trait like height varies little. In an uncontrolled playground, it varies wildly. This genetic and environmental variability creates biological noise, where patients recover or decline unpredictably, making it impossible to see a drug's true signal. To solve this, researchers must establish strict internal validity. They design a structure that isolates the factor of interest by controlling for all those outside variables. A randomized controlled trial is an engineered system designed specifically to filter out human variability, leaving only the true pharmacological signal behind. This flowchart maps the foundational structure to achieve isolation, the parallel study design. The first major filter happens here. Random allocation means every participant from the sample has an equal chance of assignment to either path. Without strict randomization, a trial is vulnerable to allocation bias. A well-meaning clinician might subconsciously assign their healthiest patients to the active drug arm and place sicker patients in the control group. Dictated randomization strips the clinician of that choice, ensuring both arms have statistically identical baseline characteristics. You also need that control group running perfectly parallel, establishing how the disease progresses normally without the new therapy. Researchers are hunting for systematic bias, any extraneous variable that distorts the comparison between these two parallel groups and erroneously influences the results. By locking in random allocation, researchers guarantee that any final difference in patient outcomes is strictly due to the intervention itself, not pre-existing traits. Structural filters handle who gets the drug, but clinical trials must also filter out psychological bias, driven by who knows they are getting the drug. Response bias occurs when a patient's outcome changes simply because they are aware they are receiving an active, promising treatment. This is problematic with subjective self-reporting. If a patient knows they are on the new therapy, they may subconsciously underreport their pain or exaggerate their improvements, eager to please researchers or validate their hope. Researchers themselves bring flaws into the room through assessment bias. This happens when the person measuring results knows which patient is in the active arm. A researcher might subconsciously interpret a borderline blood pressure reading as an improvement, favoring the treatment they desperately want to succeed. The defense against this is blinding, a mechanism that strictly masks treatment allocation from human awareness. In a single-blind study, the patients do not know if they are receiving the drug or a placebo. In a double-blind study, neither the patients nor the clinicians giving the intervention know who is getting what. The most rigorous trials use triple blinding. Here, even statisticians analyzing the final data are kept in the dark about which group is which until calculations are complete. Stripping away human expectation is the only way to ensure the data on the page reflects physical, pharmacological reality. The parallel design is highly robust, but it is not perfectly suited for every clinical scenario. For chronic conditions requiring long-term symptom relief, researchers deploy the crossover study design. The core mechanic here is sequential. A patient starts on either the active drug or the placebo. Then, midway through the trial, they swap places. Every single patient eventually receives both the active drug and the control treatment in a randomized order. To make this work mathematically, there has to be a strict washout period between the two phases. This gap ensures the effects of the first treatment have completely left the patient's system before the second phase begins. Statistically, this is a massive advantage. Instead of comparing different groups, the patient serves as their own baseline control, almost completely eliminating between-participant variation. Because that internal variance drops so low, researchers can achieve statistical significance with a much smaller sample size. It also provides a distinct ethical advantage. Every single participant is guaranteed access to the active therapy at some point during the study. Parallel models are the gold standard for testing permanent cures, but crossover models provide exceptionally tight data for chronic symptom management by turning the patient into their own perfect control. When a trial concludes and a patient shows improvement, we have to ask the critical question: how much of that improvement is actually due to the drug itself? This chart breaks down the control group response. It is actually a composite metric made of two distinct layers. The foundation is natural epidemiology. This is the baseline trajectory of the disease. Some conditions naturally improve over time without any intervention at all. Stacked on top of that is the placebo effect. This is the psychological and physiological impact generated simply by participating in a therapeutic ritual, interacting with doctors, and being observed. Now look at the treatment group response. It contains that same natural epidemiology and that same placebo effect. But sitting at the very top is the isolated treatment effect. Finding the truth requires a simple mathematical deduction. You subtract the total control response from the total treatment response. Efficacy is only proven if the isolated pharmacological effect significantly outpaces the combined force of natural healing and psychology. Through randomization, blinding, and strict control groups, a well-designed RCT achieves pristine internal validity. It creates a flawless, enclosed environment where the data is undeniable. But that rigorous enclosure comes at a cost. Extreme internal validity often compromises external validity, the ability to generalize results to the public. To keep data clean, trial populations are filtered through rigid inclusion criteria. This often results in a sample group that does not match the messy, diverse reality of the general population suffering from the disease. Inside a trial, patient compliance is monitored constantly. Out in the real world, patients forget their pills, take conflicting supplements, and experience wildly different stress levels. Researchers constantly have to balance the need for immaculate, clean data against the need for broad, realistic application. Clinical trials are an exercise in controlled compromise. We engineer out the noise of human variability to find the objective truth, while trying to maintain just enough reality to actually treat the patient waiting at the end of the process.